This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode 335, recorded on May 1st, 2015. Hey everybody, I'm Vincent Racaniello, and you're listening to TWiV, the podcast all about viruses. Joining me today here in New York City, Dickson Despommier.
Hello, Vincent.
Welcome to TWiV, Dickson.
Well, thank you so much. It's a thrill to be here.
Give me a thrill. Are you really thrilled?
Yeah, I love this show.
It's 15 Celsius.
It is.
And partly cloudy.
Not just partly cloudy. It's cloudy as hell.
Yeah, it is pretty cloudy, but it's not going to rain. 0%
That's right.
rain.
That's
Dixon, let's find out what it's like in the rest of the country.
Oh, okay.
Right? Also joining us from Ann Arbor, Michigan, Kathy Spindler.
Hi, everybody. It's 71 degrees here, and the sky looks just like Springfield of The Simpsons. Blue and the white puffy clouds.
Very
71 Fahrenheit is 22 Celsius.
So there's something going on there this weekend, right? Because I know Michelle said she's leaving town.
Well, there's graduation.
Graduation, that's it.
That's a big deal.
She rented her house out, yeah.
The better thing is that in my neighborhood, there's it's called the Water Hill neighborhood. There's a music festival where people play music on their front porches.
Oh, that's cool. That's very cool.
It's really cool. This is, I think, the 5th year of it.
Wow.
And
Wow.
it's getting bigger every year.
Also joining us today from Western Massachusetts, Alan Dove.
Good to be here.
Good
Hey,
weather?
Alan.
Good weather there? I bet it's like here.
It's pretty much like there. It's mostly cloudy. It's
Right.
There's like a slight broken, it's a few breaks in the overcast, and 59 Fahrenheit, 15 C. But not raining, and not too bad.
Also joining us from North Central Florida, Rich Condit.
Howdy, everybody.
How's it going?
Excellent. We got 78 Fahrenheit, clear blue skies, well, a couple of puffy clouds, 26 Celsius, and it's just delightful. It's a little windy, but just delightful. We're getting I thought we were going to have summer there for a few days, but we've got another little bit of spring going on, and it's nice.
All right.
Good.
I meant to tell everyone a couple weeks ago we passed our 3 millionth download of TWiV.
Hey.
3
That's
million.
cool.
And it took me a long time to download all those episodes.
Yeah, it did, Vincent. You've done a great job.
Are we still doing about 10,000 of what's our sort of...
Yeah, between 10 and 20,000 a pop. If you're Ebola, you're more closer to 20,000
Right.
Okay.
in Ebola episodes, yeah. Actually, we're above 3 million, but I changed the way we account a while ago, and we lost probably 500,000. So, but that's okay. 3 million, good. Thanks, everybody.
Yeah.
Speaking of thanks, this episode's brought to you by ASM Microbe 2016. This is a brand new meeting. Okay, it's a combination of the general meeting and ICAC, all under one roof. ICAC and ASM, the largest gathering of microbiologists anywhere.
So it's ICACasm.
ICACasm, it could be.
Right.
Could be
Good thing it wasn't being held in Hackensack.
a real, truly global event where you can find the best microbiology. Actually, I'm not supposed to say microbiology. I'm supposed to call it
Why not?
microbial sciences. Hey, because it has been decided at ASM that microbiology is an antiquated term that is not hip, and microbial sciences is more reflective. Because if you think about it, we're attracting lots of chemists and so forth, other fields to get involved with microbes, and they don't consider themselves microbiologists. So the idea is not to alienate them, and so we're calling it microbial sciences. Does that make sense?
No, but whatever.
That's right. I don't think it does either. But
Well, I've gotten all these emails about this. That's what I'm telling you.
Okay.
Microbiology
fine. Yeah,
is
you're the PR guy, right?
I'm in a microbiology department, right?
You are.
However, lots of...
No, you're in a microbial sciences department.
that's right.
Actually, it's microbiology and immunology. They changed
Yes,
the name.
yes.
What is your department, Rich?
It is molecular genetics and microbiology.
Yeah.
It used to be immunology and medical microbiology, but
Right.
things changed.
They did.
All right. Well, anyway, back to ASM Microbe. The only meeting that explores everything about microbiology. It's really transdisciplinary. And what you will get if you go to it, all of the things you would find at the general meeting and ICAC, as well as transdisciplinary sessions, special profession of microbiology track. I'm working on that one. I'm going to a meeting on July 1st to put the program together for that. And a bigger poster hall, networking, lounges. Hey, the lounges. You have to go for the lounges.
Microbial fermentation products for imbibing.
Opportunities for continuing education, etc. Over 200 sessions, over 5,000 posters, 7 program tracks. This is just a unique meeting. You have to check it out. I'll be there. It's in Boston.
Oh, it's in Boston. Okay.
When?
It's probably going to be in...
They have the
May-ish.
dates, in fact. June. It's
June.
June 2016. I'll get you the dates. I have them on my calendar.
Yeah, you ought to go, Alan Dove. Well, we'll do a podcast and you
Yeah,
can come out.
if you're doing a TWiV from there, I can make it out for that.
Yeah.
I'll be a liberated man. That would give me an opportunity, an excuse to go to Boston, visit
That'd be
my daughter.
great.
I have that it's June 16th, a Thursday, through Monday the 20th.
Roger that.
Except I'd probably have to pay money, right?
Seems that way.
Yeah.
No, you're with TWiV.
Yeah.
Yeah.
That's right. Just use your TWiV entry card.
We might be able to get you a press pass. That's what we do for Alan Dove.
All right.
Anyway, check it out. asm.org/microbe2016. Should be interesting.
I'll bet you if you go, you get a cool meeting bag, too.
Oh, yeah.
Oh, and you can get all kinds of swag at the
Yeah.
vendor displays.
Kathy got some cool stuff last
Yeah, I
night.
got the gigantic microbe E. coli.
I got pictures of you with those, yeah.
Ah.
Yeah. That was in Boston, right? That last
That
one?
was in Boston.
Yeah.
There's going to be a lot of people, right?
Oh, yeah.
Oh, yeah.
Yeah, lots of people. That's part of the idea. Cut the costs and have all those people come together, I hope it works.
Right.
We'll see.
Yes.
Big meetings, it's a certain kind of animal, a big meeting. All right. We have some follow-up. This one from JS was actually on the TWiV.tv website, and I thought merited being spoken here. He writes, I think I heard someone yawn when you listed all the responsible conduct of research topics in the special JMBE issue.
Yeah, it was probably Dickson.
Hopefully unintentional. I don't think it was you, Dickson. You would never do that, would you?
No, of course not.
So he gives a link to the issue, and then he writes, according to this 2013 piece, Dr. Oz still performs operations there each Thursday. And there's a New Yorker article that tells us that.
I'm taking my heart elsewhere.
Yes.
No, he's known as a good surgeon, by the way. That's
I'm
not
still
one of his
taking my
faults.
heart
to be
elsewhere.
honest.
Spector also wrote a blog post on the recent letter from those physicians. It's also in the New Yorker. He lamented that I didn't have a link for the Wine Spectator, but I fixed that. He says, Alan will be glad to know that we have a barley genome sequence.
Hey.
Yes.
Are you glad to know, Alan?
Yes, I am. I am glad to know, and it barely escaped my notice.
Oh.
And his jokes seemed more over the top than usual in the Ebolavirus section. You should get a drum roll cymbal crash sound.
Oh, they sell them.
I don't have to. I can get them. I don't need to. That's a good idea. We could do that.
That's probably built into
program.
GarageBand or something, isn't it?
Maybe.
But don't use GarageBand. We moved
Okay.
beyond that about $1 million.
We ought to be able to
ago.
put a button on my browser that'll just click that button.
I can put it in post, and you can be surprised. And finally, he says, "I hope you do another show with Jens Kuhn." Yeah, I hope so too. Since Jens was in the Times, he had his picture in there. He might be harder to get.
Well, that's what happens when we give people the TWiV bump.
Oh yeah.
Yeah.
All right, Kathy Spindler, would you read the next email, please?
Sure. Juan writes, Dear Alan, Dickson, Kathy, Rich, and Vincent, alphabetical order. Thanks once again for an interesting episode of TWiV, and also for choosing plant viruses as one of the main topics of the show. The digression about flowers was indeed funny, and being a plant virologist, I fully align with Dickson's side for obvious reasons.
Yay.
Among others, flowers can serve to diagnose virus infection of plants.
How about that?
To refresh Vincent's memory, I want to mention a link from his own blog.
Uh-oh.
He gives the link, dealing with tulip breaking virus. The nice patterns in these flowers are diagnostic of the presence of potiviruses, and they can be found in many old paintings from the Dutch masters.
Indeed.
Even the name Rembrandt tulip breaking virus was once proposed for a potivirus species. And he gives the PubMed link for that. although it was not accepted formally by the ICTV. What's wrong with the ICTV?
Exactly.
Oh, anyway, for Spanish-speaking listeners,
I'll
there's
tell you what's wrong with the ICTV.
This is a can
it's
of
made
worms,
up of
folks.
committees that are made up of people.
Oh, that.
Okay.
Yeah. Okay. For Spanish-speaking listeners, there's a nice divulgative article by Jesus Navas Castillo, and he gives a link for that. It is indeed in Spanish, and it's about the tulip bubble. It reproduces with permission from the Museo del Prado, which is the big art museum in Madrid, a beautiful painting by Jean Bruegel El Viejo.
Huh.
Best regards, Juan. How would you pronounce his name? Juanjo? Juanjo. Yeah.
I guess.
Juanjo. PS1. Weather info. Gorgeous springtime weather in Barcelona. Mostly clear skies, 20 degrees Celsius, 68 Fahrenheit. Humidity 67%. A bit late for tulips and cherry blossoms, though. PS number 2. When searching for links to this email, I realized the POTIvirus entry in Viral Zone needs to be updated. It is incorrectly mentioned that incorrectly mentioned there that the extra P3N Pipo gene product, quote, is expressed by a -1 ribosomal frame shift.
I knew that. I don't know why they made that mistake. Jeez.
Hey, he puts a sick
How
in there.
could
Notice
they do
that?
that?
Yeah.
He does.
However, recently it was shown that frameshift gene products in POTIvirus is derived from a mechanism of polymerase slippage, and he gives a PubMed link for that, which reminds me of the transcriptional editing as described in Ebolavirus. I apologize for the self-reference, but I wonder if this might qualify for a TWiV bump. I guess that
It's his paper,
PubMed
yeah.
is his paper. Okay.
Yeah, that's his
So-
paper.
Yeah.
Actually, this is Juanjo. I recommend that you actually contact Viral Zone directly. They're very responsive and very responsible, and they'll make the correction. They're fun to communicate with.
Cool.
Yeah, they're good guys. They have TWiV on the front page there
That's nice.
for a long time.
Yeah, tell them TWiV sent you.
That's right.
Right.
I just wanted to mention this. Yesterday, I drove past a home with a huge tree in front of it. I don't know if it was a cherry or magnolia. You know, I don't know the difference. But anyway, most of the petals were on the floor, and it was a mess. And I wanted to
A
take
mess?
a picture and show you guys.
Well, they're gorgeous.
That's
when that
probably
happens.
a magnolia.
Mess.
Yeah, that's a magnolia.
They make a mess.
real
Big
mess.
petals,
Huge.
That's,
big white
Yeah,
and purple petals.
That's
Yeah.
magnolia.
Yeah, they're gorgeous trees, but
They are.
they make a mess.
Rich Condit, can you take the next one, please?
Ramon writes, I think this clarifies CSF glucose level meanings in CNS infections. Quote, CNS infections may alter glucose transport across the blood CSF barrier, resulting in low CSF glucose level, termed hypoglycoria.
Glycorrhea.
What?
Hypoglycorrhea.
Good, thanks. Further reduction in CSF glucose levels may result from glucose consumption by WBCs in organisms, white blood cells in organisms. Reduction of CSF glucose relative to blood glucose is characteristic of meningitis caused by bacteria,
Right.
mycobacteria, or fungi.
Right.
The CSF glucose level is usually normal during viral infections, but low CSF glucose levels are occasionally observed in meningoencephalitis caused by mumps, enterovirus, lymphocytic choriomeningitis, herpes simplex virus, and herpes zoster viruses. And there are several references attached to that, and he says this is quoted from Infectious Diseases of the Central Nervous System, edited by Sheld et al. And published by Lippincott, Williams, and Wilkins. And Ramon is an MD, Internal Medicine Department Chief in Spain, in Ibiza.
Ibiza.
And he says, P.S. I wonder if a physician's point of view could be a good complement to your excellent explanation on viral basic science.
Propitious of him to ask that question, since on TWiP just days before we got such an opinion from a physician, Dan Griffin. So I see that's in
You know, having a
great.
physician as a regular
That's good stuff.
co-host would be great. You know? because we run across this stuff all the
This
time.
is all
We're really
true.
stupid
This
about
is-
it.
Well,
That's
I
true.
tell you what, Dan is a little busy to do 2 podcasts, but whenever we have a question, I'll run it by him. So I did run this by him on TWiP. I recorded. I will stick his comments in here. I don't want to play them now because I can never figure out how to hear them while I play them. But he basically agreed. Actually, this lowering of CSF glucose in some virus infections, he didn't mention that at all. We had a discussion recently about the significance of altered glucose levels in the CSF in viral or bacterial infections.
Yes.
Can you tell us what deviations from the normal glucose level would mean?
Yes, definitely. When we do CSF, there's a number of parameters that help, I guess, give us a sense of whether it's likely to be viral or bacterial. And the idea maybe to help people remember this is the bacteria are actually eating stuff, right? They're consuming. So if you think of it that way, the glucose levels are going to be down in most bacterial infections, where the viruses are inside the cell doing whatever they want to with the host cell machinery. So they're not going to be out there eating the glucose. So you usually will end up with a normal glucose levels in viral infections, a decreased glucose in bacterial infections.
Does it ever go up?
Does the glucose
level?
ever go up? It's always important that you check a serum glucose relative, because you could have a diabetic and the glucose is high and you say, oh, obviously it's viral, but it may be low relative to serum. But there really aren't any infections I know of where you're going to end up with a glucose in the CSF higher than the glucose in the serum.
So if you have someone with suspected meningitis or some CNS infection, you would do a spinal tap and look at parameters, including glucose?
I think it was actually good when you said CSF, because we're with CSF, when we're doing a lumbar puncture, when we're putting a needle into this space, we're getting the cerebral spinal fluid. Right. And we're looking for an infection involving that space rather than, let's say, an encephalitis, where it's actually in the brain. You may not see changes in the cerebral spinal fluid. You put a needle in, you draw out the fluid, you'll look at glucose levels, which are helpful. You might look at protein levels, which would go up and down a certain. You'd look at cells, you look at cell count, and you look at types of cells. Let's say the cell count is really high, and it's a lot of PMNs, polymorphonuclear cells. That would go along with your bacterial infection. I've got Dickson over here mouthing the answers to me. So if I get them wrong, it's because I listen. But if it's, let's say it's lymphocyte predominance, you have almost all lymphocytes, that would be consistent with viral or a chronic mycobacterium. So there are some subtleties there.
Okay. All right. Thank you, Daniel.
Oh, you're welcome.
All right. Alan Dove, you're next.
All right, Lee/John, that's our friend, the writer and graduate student, writes, Dear TWiVumvirate, listening to TWiV 334, I had to laugh to hear about Dr. Racaniello's lack of interest in cherry blossoms. I'm a big fan of them, and when I'm not buried under a mile of experiments, I like to go to the Cherry Grove in West Central Park to read or just quietly enjoy the grove. No doubt more than a few listeners are writing in to call Dr. Racaniello a curmudgeon, as some of you did as well, but I think Dr. Despommier had it right. Dr. Racaniello would rather look at plaques. And what isn't beautiful about a good plaque assay?
Look at you. Look at behind Dickson. There's a huge
There's a wall of plaque behind me.
Oh, good. But the discussion of Pinot Noir and virology in the episode made me realize that perhaps there's a virology story in cherry blossoms as well. Google brought me 2 viruses of cherry trees, Prunus necrotic ring spot virus, PSNRV, and cherry leaf roll virus, CLRV.
Right.
Plant viruses have the best names.
They do.
Absolutely.
They do.
These are both group 4 viruses possessing single-stranded positive sense RNA genomes, and so I think this would be of particular interest to Dr. Racaniello.
Ah.
According to Wikipedia, which I should confirm but haven't the time, co-infection of cherry trees with these 2 viruses leads to rapid decline of the tree. Interestingly, both of these viruses are segmented viruses, with PSNRV having 3 genomic and 1 subgenomic element, and CLRV having 2 genomic segments. These segments for both viruses are packaged into separate virus particles, which must co-infect cells in the host. I find these situations where a virus spreads itself across multiple particle types especially interesting because it relies on luck for the virus to get all of its genome parts into a cell. It's almost as though these viruses' infections are a cooperative project of 3 separate attenuated viruses, or 2 in the case of CLRV. Because both viruses have this multi-particle system, I find it interesting that co-infection with both viruses can cause a more severe pathology. I know nothing about the current state of research on these, whether the severity of disease is simply an effect of stress to the host or some factor of superinfection, but it seems like there's a lot to cherry trees that is of potential TWiV interest beyond just the blossoms.
Right.
Anyway, perhaps this will make cherry blossoms more interesting to Dr. Racaniello, perhaps not. But having been inspired to learn this stuff by TWiV 334, I thought I'd write in and share another example of the fact our planet is just lousy with viruses. They're everywhere. Cool.
Yeah, you don't need the blossoms to do this work.
Yes, you do, Vincent. There's this thing about plant reproduction that involves blossoms,
Yeah, I know, but
fruit.
the blossoms go away and you can get on with the work.
Jeez.
I'm not a curmudgeon. I'm not a curmudgeon.
No,
It's just
you're
they don't
a
do anything for me. Listen,
Texted.
I've been thinking about this all week, okay? And you guys didn't ask me what I like to look at.
Okay, what do you like to
I like to look at plaques. I really like a nice body of water, okay? Like the river outside the
okay.
window. I
That's
love
good.
looking at
That's
it. It
good.
relaxes
That's
me.
good. Yes.
I like waves on the ocean, lakes.
Yes.
Crater Lake is beautiful.
Crater Lake is beautiful.
Mountains are pretty cool.
They're not bad.
Canyons.
The bridge, you like looking at the bridge. I know you
Fifth
do, because
Avenue
you can
is
see
pretty
it
neat.
out.
Okay, so there are lots of other things, but just cherry blossoms don't do it. And part of the reason is that everybody likes it. So,
Oh, I see.
like my son
black.
gave me a T-shirt the other day. It's a Dropbox. It's cool. It's got the Dropbox logo. I said, "Why don't you want it?" He said, "The recruiters came to campus and gave them to everyone. I don't want to wear the same T-shirt as everybody else." So he's a chip off the old apple tree. I mean cherry tree.
I beg your pardon. I'm the apple tree.
Hey, this is a cool thing. This bipartite genomes. We should do a TWiV on this someday.
Yeah, it seems like really stupid to package your genome in 3 separate particles.
Now you're anthropomorphizing viruses with Condit.
Listen, I'm almost retired. I can do that. It's okay. All right, I don't have to write any more grants. I can anthropomorphize all I want.
No, you cannot on TWiV.
I can't? No?
I guess you're going to
Dang.
you're not going to do TWiV. That's the thing.
Oh, no, I am going to do TWiV.
Well.
Until you kick me off because I'm anthropomorphizing about
I'll just yell at you, just like I yell at Dickson.
Yeah, all the retirees in the corner.
But, I mean, it's nuts. Why do that? I wonder if there's any selective advantage to doing that.
That's a better question.
Well,
Maybe.
each viral
That's a
particle
much better
smaller.
question.
So
Yeah.
now it's packaging smaller particles, and I guess obviously
That's for recombination too, doesn't it?
it infects at a high enough frequency that it does get the job done.
Yeah, I guess so.
How does
So
it
it doesn't
get
matter,
transmitted?
if something in biology, it doesn't matter how shoddy the engineering is if you manage to reproduce yourself.
Yeah, does anybody know how it's transmitted?
Usually these things are vectored by insects,
That's
right?
correct. That is correct.
Passively. That is, they don't infect the insects. Have I got that right?
Sometimes.
Yeah, I think in many cases, if I'm remembering from ASV where that guy gave that cool talk about plant
I mean, they come along with
viruses
pollen and stuff like that.
getting in via nematodes, right?
Right.
Remember that?
Anything
They inject
that'll-
them. I don't think they may not replicate in the vector.
They could go through the stomata, but not in the plant, not in the petals.
Well, we'll get someone.
No, I think
knows this.
typically they get in through wounds of some sort or another.
Okay.
Okay, and I
Yeah,
think
yeah.
they're mostly vectored by insects. Well, maybe we'll get some email about this.
we could.
We should have a plant virologist on the show sometime.
We should have a plant virologist.
You know,
on
a
the
long
show.
time ago, if you listened to like TWiV 2 or something, I did suggest that we do plant viruses at one point just to make an enumeration.
I think we've suggested about every
Every time we do this, we never follow up, though. Come on.
Well,
Let's follow up.
We've had a few plant virologists on over
Yeah,
the
yeah,
years.
we're
If you go
moving
back,
in the right
you
direction.
will see them.
Marilyn Rusink.
Marilyn Rusink was on, and Jack Morris, is that his name, at Nebraska? He was on. And we'll have more. I actually tried to do one recently, and it fell through.
Oh.
But we'll have more. All right, Dickson.
Claudia writes, "Hi, Vincent." She's just talking to you, Vincent.
No, she's
not
talking to
anybody
you.
else. She's talking just to Vincent. I am so moved by your response to the patient/advocate input regarding TWiV podcast 329 and grateful for your continued commitment to the ME-CFS conversation via TWiV, but I have to say that I am also a little embarrassed. My letter in response to TWiV 329 was written with much less eloquence than Jeannie's and obviously contains more emotional bias and a harsher tone towards the hosts to whom I incorrectly referred to as guests in my letter. Because this patient population has been ignored and mistreated for so long by government agencies as well as the medical community, media, and society at large, it is all too easy to allow feelings of anger and resentment to sneak into our more pure efforts of advocacy, and I apologize for having fallen into this trap. I hope you and the other hosts are able to receive the many scientific messages and personal experiences contained in my letter, in spite of the colorful descriptors I used about Dickson's and Alan's comments on that's why you had me read this on podcast 329. Having said that, I leave it to your discretion whether or not to treat my previous note and attachment, but I would hope that if they are read, it is because they add some value to this conversation. I am again attaching my previous full letter for your convenience, but after fact-checking with Jeannie, I decided to make a slight change in my report of the worldwide prevalence. The number in my original document, 17 million, reflects an extrapolation based on the U.S. numbers, and that includes mostly undiagnosed people, so I left it at millions. The change can be seen in reviewing all the markups in the document. By the way, the MECFS paper linked below just came out yesterday, but it is behind a paywall, which you can probably access and perhaps discuss on this week's TWiV. The first author is again Maddie Hornig, and in this small study, she again demonstrates cytokine abnormalities, this time in the CSF of the MECFS subjects compared to those with MS and no disease. Too many M's, too many S's, too many C's, but I'll get back to that. And no disease, supporting the prior plasma immune signatures in her other recent study. By the way, what is the number that she changed? Do you know what that was downgraded to?
I forget what the
No, you should say it now because she wants.
She leaves you with. She left it at millions.
It was from
Yeah, but she
17 million.
changed it.
Now she just calls it millions.
Yeah.
Okay. All right. So it's an approximation now.
So we actually did. I mean, we read her original letter. We didn't
Okay.
read the entire appendage, right? We paraphrased it because we had so many that episode. But it's online. It's there in the letters for TWiV 329. Yeah.
Okay.
All right. Thank you, Dickson.
You're welcome.
So this paper that she refers to is in a Nature Family Journal, Molecular Psychiatry, and the authors are indeed Maddie Hornig as one of the first ones, and the last author is Ian Lipkin.
Mm-hmm.
Right.
Right. We might do that at some point. Sort of, maybe in a what is it called? Snippet. Thank you.
Right. I can just read the final sentence of the abstract. Our results indicate a markedly disturbed immune signature in the CSF of cases that is consistent with immune activation in the central nervous system and a shift toward an allergic or T helper type 2 pattern associated with autoimmunity.
Mm-hmm.
Oh, that's very
And I
interesting.
find it interesting that they did what they did in this study with they looked at cases and also healthy people, but then also 40 subjects with multiple sclerosis.
Mm-hmm.
So, which may not be the best comparison population, but at least there's a comparison population in there where you know something's wrong.
Right.
So it's not going to be just compared directly with healthy.
Mm-hmm.
You've actually got another control in there. I think that's good.
All right. We have a 15-second sit rep.
Yes.
Go.
So we're going to do a paper about a new Ebola vaccine. So it is appropriate to do just a quick sit rep, which is that in the last week there were still 33 new confirmed cases of Ebolavirus disease. 22 in Guinea, 11 in Sierra Leone, and Liberia has been clean for weeks. So we are not totally out of the woods yet. The bottom line is a total of 26,312 either confirmed or suspected cases and 10,899 deaths, and we're still working on squashing it completely.
Good heavens.
All right. Thank you, Rich. Just out today, the Morbidity and Mortality Weekly Report on possible sexual transmission of Ebola virus in Liberia. We talked about it last time, but this is the complete report, in case you are interested. And again, a young lady, a 44-year-old woman, developed a fatal Ebola, and the only risk factor was having sex with this man who had recovered. They were able to to identify viral RNA by PCR in semen, but not virus, infectious virus. And they talk all about it in this article. And this young man also had intercourse with another woman, and 3 to 5 times between the last week of February and March 15th. All right, and she seems to be okay. So I just want to retract my earlier statement that nobody who recovers from Ebola is going to want to have sex, because he obviously did.
Right.
With 2 different people and multiple times. So I'm obviously living on another planet.
Well, I thought what you said, or what I inferred from what you said, was that while they were in the midst of the disease, they probably weren't going to feel like it.
I figured even after they'd recover, they'd have lingering. I mean, my understanding is you have lingering effects for a long time, but I'm wrong. And a lot of people yelled at me and said, "What do you know?" And I said, okay, you're right. I don't know anything, obviously, because this fella was quite active. And the recommendation is now, if until we know more, we better wear a condom, because who knows what's going to happen.
So in one of the news, I haven't read this MMWR report. In one of the news write-ups that I saw, there was something about the sequence of the virus in the woman being identical or similar to the sequence of the virus in the man.
What sequence there was identical or very close to what was in the man. And other than that, in Liberia, there's sort of been one thread, clade, whatever, of Ebola, and the sequence that she had was different from that and more like his. So it seemed like she couldn't have gotten it from something else in Liberia.
Right, okay. Mighty suspicious.
Now this, did I miss it in this report, or do they not say what ended up happening to her?
She died.
They don't say, I don't think.
Did she die?
Yeah, the 44-year-old woman from Monrovia, she died.
Ah, okay.
We said that last year, yeah.
I didn't see it in the MMWR.
They didn't mention it in MMW. It seemed like a rather significant fact.
Yeah, it does seem like a rather mortal kind of event.
It was in one of the links that we had last time. So they say that they got 20% of the genome from him, and it closely matches her sequence. So it's probably not identical, but close, right?
Right.
All right, so that you can go read that. I wanted to talk about a paper published in Science called Ebolavirus whole virus vaccine protective in primates. And this is, oops, I'm on the wrong paper. I have to open it from I didn't open it, Dickson.
Vincent, can I just add the fact that I didn't know which paper you had selected for this show, so I inadvertently...
Yeah, why would you know? I only sent you the show notes a week ago, right?
Because I didn't have time to look. So I inadvertently brought in... What article did I bring in?
Yeah, this one.
Did I really?
Well,
did I do that?
Normally I would
How could I have done that? I don't read.
Normally I would be impressed, but I'm more disturbed that you don't look at what I send you. Never. You have never looked at a single show note until the day of TWiV, and like 5 minutes before
I like surprises.
You're like, Vincent, how do I open this show notes? Every friggin' Friday you do the same thing. All right. The first author is Andrea Marzi, who is from Rocky Mountain Labs in Hamilton, Montana, and who I met last summer. And she told me all about this work. And the last authors are actually a bunch of people from Rocky Mountain and University of Tokyo. We got Yoshi Kawaoka, the last author, Heinz Feldman, Gabrielle Neumann, Friederike Feldman, Leslie Schubert, Peter Halfman, Lindsay Hill, Batorsky, and I think I got everyone. Mm-hmm. So this is interesting in the sense of how the virus is made. I believe we have talked on TWiV about previous Ebola virus vaccines, the chimpanzee adenovirus recombinant, right? We talked about that in the use of 2 different serotypes to give really durable protection. But they say here, you need a lot of that virus, so it may not be the best. We also, I think, talked about a VSV recombinant where the glycoprotein is replaced with that of Ebola virus. And that is also looked good in macaques. And they say here, but maybe there's safety concerns. So we need another vaccine candidate, and here it is. What I like about this is the way this vaccine was prepared. So previously, these individuals had shown that you can delete the VP30 gene from the viral genome, and you can produce particles in a cell line that produces the VP30 protein. you'll now have particles made that that are produced from the cell, and when those infect the new cell, they're no longer infectious because they don't have the gene, although they got assembled and so forth properly. So, and that actually is cool because you can work with that virus, which is not infectious, under BSL-3 conditions. Okay, and this is the vaccine that they're exploring in this paper. So you have a full-length DNA copy of the Ebola genome in a plasmid. You cut out the VP30 sequence. I believe in the previous paper they stuck in either a neomycin resistance gene or green fluorescent protein.
Mm-hmm.
Right?
And interestingly, that previous paper
Yeah.
was like 2009.
Yeah.
So they've been working on this since way before this epidemic.
Yes.
Yep.
Okay. It's not like they just scrambled to put
Oh,
this
yeah.
together
Yeah,
in
for sure.
response to the epidemic. And we ought to remind people that this is a negative sense single-stranded RNA virus. And so it's transcribed initially during infection by a big polymerase that's called the it's the product of the L gene that transcribes the genome, goes along and starts and stops and produces a family, 1, 2, 3, 4, 5, 6, 7 different transcripts that encode all of the appropriate viral proteins, including the stuff that ultimately encapsulates and transcribes the RNA, and that consists of NP, which is the nucleocapsid protein that wraps it all up, and the L protein in much smaller amounts, which is the polymerase, and 2 other proteins that help in transcription, VP35 and VP30. And this VP30 protein is required for the transcription, but not the other type of RNA synthesis that goes off of this, which is the synthesis of the full-length genome. So without this protein, the virus can't do transcription well. Though it occurs to me, if you grow this thing on the complementing cell line, you're going to make particles that actually contain the VP30 protein.
Sure.
So those particles will get into the cell, and they'll probably do a little bit of transcription.
Yeah.
But they can't make new VP30 or new polymerase, so they couldn't really amplify that transcription or replicate the genome to any significant extent. So the infection is going to stall real early.
Right.
Yeah, so in that previous paper they passaged this virus 3 times on cells that don't produce VP30, and they never detect any viral antigen.
I'm surprised it's even BSL-3. I mean, I think I get it, but I'll bet you in the long run this winds up being BSL-2.
Maybe.
Now, the whole process is BSL-3. So you can grow the viro cells that express VP30, the complementing cell line, and culture the virus and grow up as much of this as you want at BSL-3.
Right.
I guess it would make sense that that's
because
BSL-3.
it can't get out.
Well,
Really, it's trapped in these cells.
They actually wondered if RNA produced from the VP30 gene in the genome would somehow recombine into the virus, but they didn't see that, and so they passed it 7 times, and they didn't see any rescue of infectious virus.
So they treated it with hydrogen peroxide just to kill it.
No, not yet, Dickson. That's later.
And
I was reading the abstract. Come on.
One important thing too that they showed in a previous paper is that when they grow this virus on the complementing cell line, they get out particles that look exactly the same as a wild type infection.
So this is a cool use of a complementing cell line, and as Rich said, the particles look good, titers are high, so it's really nice.
And the ability to grow this at BSL-3 makes a massive difference if you're interested in manufacturing a vaccine.
Absolutely.
Because that's orders of magnitude. I mean, it's still a significant containment barrier, but it's orders of magnitude less expensive than building a BSL-4 GMP facility to make this thing.
Oh, yeah. Yeah, that's a big problem. And I think that's why they did this initially. So they use this vaccine candidate has already been in mice and guinea pigs, and so these are animals where you need to adapt Ebolavirus to really kill them and be lethal in them. So it's not a great model, but this vaccine protected them. And so then they moved in this study to cynomolgus macaques, and this was done at Rocky Mountain. They have a colony there, and they can do BSL-4 infections of them. So they do intramuscular immunizations with a variety of controls. There's just medium. They give 1 dose of this virus, or they give 2 doses 4 weeks apart. And then, as Dickson said, they also include a hydrogen peroxide or gamma irradiation, because they say people may be worried about using this virus without further, even though they've shown it's not infectious, they can imagine that there's some concern, right?
Yeah, okay, go ahead.
But what, Dickson?
No, it's too far ahead. I'm going to wait now.
Good. Excellent.
Sorry.
It's okay. Anyway, so then they challenge, small numbers of macaques because they're expensive. But anyway, the mock vaccinated with just medium, when they get challenged. What's the challenge? IM? I don't remember. Does anyone remember?
I don't remember.
Let's see. I'll scroll back up. Challenge. I'm just want to know.
I think it's going to maybe be in the materials and methods.
It's IM. It is IM.
It's IM. All right. So the vaccine, the medium receiving monkeys, they all died. 0 out of 4. They had to be euthanized at day 7. The ones that received 1 dose of this Delta VP30 virus, they're all protected. 100%.
But as we will see down the road,
right,
there's some issues.
they get 2 doses 4 weeks apart. They're 100% protected also. Hydrogen peroxide treatment, 100% protected. Only 2 months.
That's of the virus.
Of what? Of the virus, not the monkey?
Yes.
Right. Unless you want a
They're
blonde
blonde
monkey.
and
You want a blonde
protected.
monkey? Go for it.
Oh, boy. And then gamma irradiation.
Yeah, this is my question.
Is nobody was protected. They
Why
have
is
to-
that?
Well, you'll see Dickson actually. They actually
See,
go into it.
I'm-
But, offhand, what would you say is the reason why?
Well, because it didn't affect their genome at all.
no.
Gamma radiation is supposed to knock apart.
No, Dickson, they're not protected.
No, I understand that.
They're challenged with an infectious virus.
Yes.
So this virus was wrecked antigenically, so it didn't induce protective antibodies.
Oh, I see what you mean. And then-
Right.
So the gamma
I got it,
radiation is
got it,
probably
got it.
screwing up the
I got it,
particles,
okay,
including
fine,
screwing
fine,
up the
fine.
proteins.
Fine.
Interestingly, this hydrogen peroxide treatment is fairly new. The
right.
paper they refer to is 2012.
Yeah.
And the hydrogen peroxide apparently is pretty specific for nicking the genome and not messing with proteins. So it leaves the particles in pretty good shape but trashes the genome. That's in contrast to older techniques, techniques that are, they're still currently being used for inactivation, which is usually formaldehyde, right?
Or
Yeah.
heat.
Yeah, formaldehyde. I don't know of any vaccines that are heated, but I know formaldehyde is one of the IPV and so forth.
It just cross-links the proteins, though.
The
doesn't it?
the interesting thing here is that the gamma irradiation was developed to help disinfect BSL-4 labs, right? So this is a
Sounds like it
whopping
works pretty good.
massive dose with no regard for antigenicity, right? Because you just
Right.
want to clean up your facility. So that's
So
that.
this trashed both the proteins and the genome itself?
Well, they didn't actually look at the genome. They looked at the antigenicity, as we'll see in a moment, but it could have also trashed the genome, right?
Yeah.
So these animals, they did take blood at different days post-challenge, and the ones, of course, who were vaccinated with just medium, they develop a nice viremia of 10 to the 7th, 10 to the 8th, TCID 50 per mil, lots of virus, all 4 of them. And in this group that only received 1 dose, one animal developed a lower viremia, 10 to the 4-ish peak. So, and then if you give them 2 doses of the vaccine, there's no viremia. If the hydrogen peroxide treated vaccine, no viremia. And
that was also a 2-dose
It
protocol.
was a 2-dose. And then the gamma irradiated, all 4 animals had viremia.
Yeah. It's,
So I really like this set of experiments because it actually, I think in particular when you're going into non-human primates and you have to craft your experiments pretty carefully, a question is always 1 or 2 doses.
Yeah.
And I'm
Yeah,
glad
yeah.
they did both, and I'm glad they got data that really suggests that you're better off with 2. And
The doses are not huge. 10 to the 7th focus forming units, right? And if I remember, I just looked at the chimp adenovirus Ebola vaccine. It's 10 to the 10th, 10 to the 11th.
Right.
It's
that
a
was
lot.
one of the problems they cited with that.
Yeah, so this is a little better. It's many orders of magnitude less, so that's better.
But again, with a 2-dose vaccination program, if this were to be applied to people, the follow-up for the second dose is tougher than just getting everybody to do it once.
So it's nice to know that you get partial protection with one dose.
Yeah,
Yes.
no, that's true.
More than partial protection, at least with this small sample size.
Well, yes. I mean, but in the one case, you do see some viremia happening.
In one animal, right,
Yeah,
and
in
another
one
animal
animal.
had a temperature.
Right.
So, yeah.
So it suggests you really do want the 2 doses. But to get to Dickson's point, in the settings where this is happening, you go to Liberia and everything's falling apart because there's an Ebola epidemic. At least if people have gotten one shot, then they're a lot less likely to die of this and probably a lot less likely to spread it.
Yep.
Mm-hmm.
They also looked at serum antibodies in these animals in the different groups at 2 weeks before challenge and then at the day of challenge.
Right.
And, the one that gave the highest antibody titers consistently was this 2-dose of the untreated VP30 deleted virus. And the hydrogen peroxide gave reasonable levels of antibodies as well, a bit lower.
Actually, if you look in the scatter in the data on day 14, it really, I don't know
It's right
that
in
there's
the
much
middle there.
difference between the hydrogen peroxide treated
not.
and the
Because on day 0, it's pretty similar, right? Yeah,
right.
the two.
But clearly the boost is having an effect on the serum antibody titers, which makes sense.
These gamma irradiated viruses induce no antibodies. Apparently.
Yeah, they're really a mess.
Really there. Then the other experiment they did, which addresses your question, Dickson, what does gamma irradiation do? They take a panel of monoclonal antibodies against the glycoprotein and then simply do a binding assay
Yeah, I see what you mean.
against each, either gamma irradiated or hydrogen peroxide irradiated.
Right.
And, the gamma irradiation trashes a lot of the interaction. Not every one, but it trashes a good number of them. And then one, the glycoprotein is way more reactive with gamma irradiated than with hydrogen peroxide irradiated. So the suggestion is that some new epitope is now really exposed as a result of this. So it's quite different. So anyway, this is pretty neat.
Yeah.
Yeah.
And yes.
I have something. So when they talk about the results with the antibody to GP, which is in Figure 2, they say these data indicate that immunization elicits an antibody response to GP that is important for protection against Ebola virus infection. Okay, maybe I just answered my own question. The next sentence is, they've seen a similar correlation between GP-specific antibody response and protection with other Ebola virus vaccines. The reason I'm asking this is because mostly they say that because the pattern of the antibody response correlates with the amount of protection that they see, that says that GP is important, one of the correlates of protection. But then on the next page, and in the supplemental data, they also look at antibodies to NP and VP40 because the difference of this vaccine from the others is that it's the whole virus, so there's other proteins there. Yep. And they see, to my reading of it, the same kind of correlation among the groups, and yet they say the importance of NP and VP40 antibodies to protection is not yet known.
Right.
So to me, it's like, well, you can say it for GP, but you can't say it for these other two. But I guess it's based on the additional data from other papers.
It's interesting because if I believe I've got this right, the natural infections to measles and mumps, which are viruses in the same order of viruses, one of the predominant antibody responses you find is to the nucleocapsid protein. That's always been a mystery for people studying these, but it indicates that some of these other proteins could have a role certainly in the immune response and maybe even in protection. So there may be something to this, using the whole virus instead of just something that expresses the glycoprotein. I could be dead wrong here. I'm making this up, but there you go.
Do you think it'd be worth in a rodent, for example, making a particle without the GP and seeing how it compares?
That would be interesting.
I mean, in terms of making it, you might as well have the whole thing, right?
Yeah.
It doesn't have to help.
Sure.
But in terms of understanding which part is important, that might help. I don't know. It might be done.
The other thing they mentioned about this is that these particles are still going to have their nucleic acid in them.
Yeah.
And if you treat them with hydrogen peroxide, evidently you don't really mess up their nucleic acid. So that could be important for eliciting innate immune responses as well.
Yep, absolutely, for sure.
So, actually, the hydrogen peroxide does mess up the nucleic acid.
In
say
what
here
sense?
Causes
right. I did have that backwards. Yeah,
single-stranded and double-stranded RNA breaks in single and double-stranded RNA.
You still could
DNA,
have enough
DNA.
RNA to. It depends how many breaks, right?
Right.
Could have enough to still stimulate sensors, innate sensors.
Right. The RNA is there, and probably something's going to happen to it when it gets into the cell. But it's not. You've rendered replication incompetent.
Right. But certainly, I would imagine it not sufficiently to necessarily trash the response to the intercellular response to the nucleic acid.
Yeah.
Right.
So this may end up. I guess next would be a phase 1 in people, right? There's got all the animal data, all the preclinical is done. So this would be interesting. This looks very promising, and
I think so.
to me,
Absolutely.
right? Maybe more so than some of the others. Okay.
So I have a question. Just
Dickson,
back to
now since you had picked this, what do you think?
Well, I thought I picked very well.
Okay.
I see red flags every time I pick up science and I see an article about viruses, and I have another one that I picked too, but that's for another time. But I had a question about Rich's recounting of the current epidemics. And that is there a histogram presentation of the data? Because when I asked our physician on call, Dan Griffin, to update me with regards to the incidence and prevalence of Ebola in the 3 countries, he showed me a graph that continued upwards and upwards and upwards.
Yeah, that's cumulative case.
Yeah, cumulative is a lot different than showing it the way they're showing it here, though. Yeah. It's waning. There's no question about it.
Oh, yeah.
Yes.
Well,
it's waning. Except
for the it's we are asymptotically approaching wiping it out. Okay,
it could flare back up.
If you look at a histogram that goes back many more months than this, there's a precipitous drop at some point.
That's
but
right.
But it's at the bottom here. It's really difficult squashing these last few.
Indeed.
Yeah, keep on scrolling down to figure 3.
Figure 3 and 4 have nice graphs,
so
Dickson.
I'm going to go back to him.
He was
using his cell phone to show me this data.
Well, that's okay. That's not a problem. But
he forgot to scroll to that part.
Yeah.
We have to have a talk.
Go have a talk with Dan.
I'm going to do that, but not right now.
Okay. You can go. It's okay.
Anyway, for the listeners, the figure 3 that we just referred to is on the SITREP page.
The SITREP.
That's it.
All right.
Should have Wolf Blitzer do that, though,
Snippet.
don't you think?
Who?
Wolf Blitzer.
I don't know
He's always from
he
the
is.
Situation
Room.
It's CNN.
Okay. We have a snippet, 2 snippets. There are 2 papers about the evolution of Ebolavirus. One of them is called The Mutation Rate and Genotype Variation of Ebolavirus from Mali Case Sequences. And this was published in Science. The first author is Honan. The last author is Sau. And Heinz Feldman is also on this one. And the second one... oops, I don't have it open, Dickson. I'm going to have to open it here.
Don't look at me.
I'm not. I'm
I'm
actually facing away from you. The second one is in virology, and this is an interesting subject, why it's in 2 different journals. But anyway, Ebola virus is evolving but not changing. No evidence for functional change in Ebola from 76 to the 2014 outbreak.
Amazing, isn't it?
So in both papers, they look at a variety of viral sequences in slightly different ways. The virology paper is very good. They look at the numbers of parameters, including, the where in the genome the synonymous versus non-synonymous changes are, right? Remember, the ones that change the amino acid versus the ones that don't. They look at the sites in the proteins where these changes are occurring and try and see, because a lot of these proteins, their structures have been determined, so you can see if they're in ordered or disordered sites, looking at areas that interact within the protein, and try and see if these have any possible functional consequence. So the virology article concludes that the changes have, none of the amino acid replacements lead to identifiable functional changes. They have minimal effect on protein structure, neither stabilizing nor destabilizing, not in regions associated with known functions, tend to cluster in poorly constrained regions, right, flexible regions, disordered regions. We find no evidence that the difference between the current and previous outbreaks is due to evolutionary changes associated with transmission to humans. Instead, epidemiological factors are likely to be responsible for the unprecedented spread.
You mean, Vincent, there's no gain in function occurring in nature?
No, apparently not.
No.
Darn.
It hasn't gone airborne.
Darn.
Hasn't gone airborne, Glenn Beck.
Nor do ferrets transmit it to people.
And then the other paper, so they start out by saying previous analyses based on limited available sequence have been used to suggest that Ebola is mutating more rapidly during this outbreak, potentially as a result of sustained and large-scale human-to-human transmission, which has raised concerns about increased virulence and transmissibility, which of course always happens. Anyway, they calculate the mutation rate is the same as in previous outbreaks. Again, these mutations are occurring in regions that encode, disordered regions of proteins, not functionally relevant. And they conclude that the epidemiological and case management data do not support increased virulence in humans during the current outbreak. It's doubtful that either virulence or transmissibility has increased despite the extensive and prolonged human-to-human transmission.
Yeah.
Go ahead, Kathy.
I was just going to say, what I found interesting is I didn't realize that pretty much all the sequence data that we had so far is from March 2014 and June 2014. And so in this science paper, they used these patient samples from Mali that were from October, and basically they show that they're really not that much different from the ones isolated in June.
It has not changed very much. And the thing is that's quite interesting is that, I don't know if you guys remember, there was a very high profile, it's a science paper from Pardis Sabetti's group at the onset of the outbreak. They had recovered virus from a number of individuals and done deep sequencing, and they had originally claimed early on that there was some evidence for increased mutation rates, and they say in this paper that's simply not the case, and that got a lot of people frightened. I just want to remind everyone that on September 9th, 2014, in a op-ed piece in the New York Times, Michael Osterholm wrote that virologists are not wanting to discuss the possibility that Ebola could mutate to become transmissible. And I want to read what he wrote. The current Ebola virus's hyper-evolution is unprecedented. There has been more human-to-human transmission in the past 4 months than most likely occurred in the last 1,000 years. If certain mutations occurred, it would mean that just breathing would put one at risk of contracting Ebola. Infections could spread quickly to every part of the globe, like flu. Now, it is hyper-mutating or hyper-evolving. This is simply wrong. He had no data to say that. Of course, it's mutating all the time, but not leading to functional changes, and he assumed that these would be. So I would just like to say that these two papers put to rest the suggestion that he made that it would be changing to an airborne virus. Yes, Dickson?
No, I was just going to suggest...
Why were you waving at me?
Well, because I heard someone rustling papers, and I wanted you to say something about that, but maybe I will.
I'll just cut it out in the post.
So, let me get this straight. Dr. Osterholm is not a virologist? Is this correct?
He's an epidemiologist.
No. Is he a virologist? Let me ask you that first.
No, he's not a virologist.
He's not a virologist, but he's making virological statements.
That's correct.
Right.
And so we're calling him out on that. And we're saying the evidence by virology, by epidemiological virology using sequence data, is absolutely opposite of what this
right.
person
And first
said.
of all,
do I get the gist of all this?
That's
right.
correct.
The data.
Okay,
the data
fine.
for
I just
what
wanted
he
to
was
clarify that.
The data for what he was arguing back in September did not exist
Right.
at the time. So he was arguing, he was talking into his hat.
He was
just
speculating and fear-mongering.
just speculating.
Oh, I see. No, he was. That's called fear-mongering.
And now we actually have the data,
Right.
which prove that this was in fact not going on at all.
Right.
Which was kind of what we suspected anyway.
Right.
But see, he published this in a very high-profile place. Lots of people saw this, and the press picked it up. It went over and over and around, and even Glenn Beck, who is not a virologist, started saying he's going airborne. Remember, we talked about that. So I think this was a bit irresponsible, right?
Yes.
To do this.
In hindsight, you'd say, of course, but not at the point of the maximum attack rate. I don't think you would say that.
Dickson, we talked about this back then.
No, it's why the public was willing to accept his view then, because they were afraid it might spread to this country.
But I think it was an irresponsible thing to say at that time.
Totally agree. I know. I agree with that part entirely.
Because you can't go leaping ahead of the data when everybody's terrified.
Correct.
And I would argue, Dickson, that the idea that a virus will change its mode of transmission is quite rare and far-fetched, and it wasn't worth for him to put this in the New York Times.
Extraordinary claims require extraordinary data, and in this case, there weren't any data.
That's right.
And so this was out of line at the time.
Sure.
And it's now we see just how far out of line now that we do have the data.
Exactly.
So I like this one virology paper. The authors said, It's not the virus. It's epidemiological parameters. Why this was an intense epidemic,
human
right?
behaviors,
all sorts of factors it got started a long time before we picked it up. They weren't prepared, etc.
Yeah,
and but everyone wanted to blame the virus early on, and now we can't. Okay, that's the bottom line.
So this is irresponsible. And I would point out that this is one of the figures involved in the original dust up about gain of function research as well.
Yes.
Right?
Oh yeah, he was on the NSABB when the Fushier Kawaoka
So
story
I would
broke.
call this episode "The Epidemiologist Who Cried Wolf."
Wow.
Period. And you take it from there, folks.
Oh, of course, Dickson. When the phone calls come, will you take them?
Sure. I'll just forward them to you.
You take all the Vertical Farm phone calls.
I do. I take them all.
Kathy, did you have a snippet?
Oh, I did have a snippet. This was pointed out by someone in my department. The director of the National Institutes of Health Institute, that's the General Medical Sciences, NIGMS, had a very interesting article that he wrote in the Molecular Biology of the Cell. And he basically described that since taking the position as director, I think he said it was about a year and a half ago or something, that when people ask him what his job is, he says it's to make sure that we're maximizing the scientific return on taxpayers' investments. And he describes a number of things. I think it's just a really well written article, how a diverse investment portfolio is a strong portfolio, both with financial investments and biology. And so having diversity at all levels, from the kinds of science to where it's done to the backgrounds of the people doing it, is important. And he's got several pieces of data in here. And the one that really caught our eye was that if you had an established investigator who has 2 R01s, let's say, and is therefore getting $400,000, they're each 8-module grants. If that investigator got a third R01 and his total direct costs went up to $600,000, that for that additional $200,000 in grant money, you would get on average 1 additional research paper. But if you gave $200,000 to a junior investigator who had no other funding, that you would get 5 papers. That's what the data say. And so his point is that having more people maybe getting smaller amounts of money or smaller sized labs is something that they're considering at general medicine and might be worth thinking. He also offers caveats to the analysis, too.
You know, we've talked about limiting the number of NIH grants, right? Some people have more than 4. Mm-hmm. And maybe you shouldn't have more than 4 or 3, whatever the number is.
These data are pretty damning. This is an open access paper, by the way, and the figure 2 from the paper, where they looked at at lab productivity by lab size, measured in number of grants and direct costs. And it goes up and then levels off and actually starts to go down at the higher level, at the larger labs. So it is very clearly showing that good science does not scale.
No.
That on average, what happens is you get above a particular size, and then the PI, probably because they're so busy writing grants and not paying as much attention to the lab itself, productivity falls off. So you're actually much better funding more people at lower levels than in making the rich richer.
Remember this guy we talked about a while ago who has 11 R01s? They didn't make any effort to limit that. I mean, that's absurd.
I have another comment to make, and that is if you start measuring productivity for scientific research based on numbers of publications, you're a fool. You're an absolute fool.
Well, there is that, yes.
That's crap. That is absolute, total, 100% crap.
But the problem, Dickson, is
I know what the problem is. Alan, you don't have to tell me what the problem is. I know what the problem is.
It's not 100% crap.
I suffered under the stings and arrows of that paradigm, and I was in a study section once where we reviewed a grant, and the PI said, for every $10,000 of grant money you give me, I produce a paper.
Well, there's one example, Dickson. Come on.
No, it stuck in everyone's mind, and it said that's the gold standard that we want to judge grants.
How
by
else you
from
gonna?
now on.
That's sad.
It's very sad. It's of course very sad because after a while,
what's your metric, Dickson, for
how many citations other people cite your paper?
Well, it's a paper in the end, Dickson, right?
No, but it could be an obscura journal,
Okay.
or an obscura idea. The Watson and Crick original article that appeared is a very short, concise article. It's one of the most quoted papers in the world.
But those are seminal articles, Dickson.
Well, that's what we're looking for, aren't we?
No, we're not. We don't all need to publish seminal articles.
I want to publish seminal stuff only.
Okay.
Minus the Ebola virus in the seminal.
I thought this was a really nicely written article, and it seems to me that the bottom line, and it's more than a suggestion, is that the most productive way to dispense the funding is over a large number of laboratories, favoring in as much as possible young investigators. And it occurs to me that also accomplishes a geographic spread. And there's more to this than just producing science. There's training scientists. And by, spreading the money into lots of young labs throughout the nation, you make the training accessible to people as well.
Take it easy. Vincent nearly broke our studio. So
I agree.
So,
I think
Richard, you don't know which my pick of the week is, and you won't find out right now because we're not finished with the show yet. But when you do hear my pick of the week, you'll be amazed at the synchronicity of these ideas.
So I agree. This is great. I think spreading out the money is a great idea. However, I do think his last paragraph is an entire cop-out. He basically says, we know there's a big problem, but I'm optimistic that we're going to fix it, and doesn't say how. Okay? So I think if spreading the money is the way to do it, or one way to do it, that's great. But he should have ended it a little better than this. But I think it's great.
Wasn't it last week that we pointed out this website where you can now write in and offer your opinions on how to fix the system?
Right.
That was my pick last week. So
Up until May 15th, right?
That's right.
Yes.
All right.
So hopefully John Lorsch will write in. And, well, he probably has more say than would go into the form.
He can write in through different channels.
Yes, he can just write in and sign it.
There you go.
Okay, let's do a couple of emails. But before that, I would like to tell you that this episode is sponsored by ICAC ICC 2015. This is a combined meeting that is being put together by ASM and the International Society of Chemotherapy. ICAC and ICC. They're going to have a meeting together this year. It's September. And it's going to be a place where clinical microbiologists, ID physicians, researchers, and pharmacists talk about antibiotic resistance, antibiotic development, and antibiotic stewardship.
So this will be ICAC-IC.
ICAC-IC.
By the way, just as a side note here, someone published a recent article showing that the Yanomami, they're one of the most isolated peoples of South America. Their gut tracts, their microbiome contains antibiotic-resistant genes in some microbes. And they've never been exposed.
They exist in nature, Dickson.
They do.
We know
do.
that. They're ancient also.
They are.
You, at this meeting, ICAC, you can choose from over 110 sessions and 19 courses about drug development and ID research. You can earn continuing education, 32 3/4 hours. There'll be over 6,000 professionals there you can hang out with, talk about diagnosis, prevention, and treatment of infectious diseases. And, of course, go to the exhibit hall. Over 70 companies will have their stuff on display. The abstracts can be submitted. You can also register through May 21st. Go to icac.org/icac-ICC. Find out more about that.
So there's ICACasm in June in Boston. And
2016.
Next
2016
June.
Yes.
Right. ICAC comes first, which is in
Correct.
September in
Right.
San Diego.
And this
Right.
summer we have just plain old GM, the last plain old GM as far as we know.
In
Just
New
as-
Orleans.
New Orleans. Are you going, Kathy?
No, I'm not.
I'm not either.
A schedule conflict.
All right. Did anyone not read an email in the beginning? Did you not read one, Kathy?
Everybody did.
Everyone did?
I wrote
Everybody
down the
did.
list. Yep.
Wow. So
Except
let's
I'm not sure if you did, but
I did.
the rest of us did. Okay.
I
Yeah, I think Vincent started off.
let's start off with Kathy. Can you take Justin's, please?
Sure. Justin writes, Dear TWiV hosts, I love the show and really appreciate what you guys are doing. I've been tempted to write in on a number of topics, but I've restrained myself because you get so many emails. My ears do perk up whenever the topic of careers for recent PhDs and postdocs is under discussion. It is true that career options for scientists have become more limited and the market is more saturated than it has been in the past. Being a PhD student myself, this is something I often think about. Despite the challenging career outlook, working on a PhD has been by far the most rewarding experience of my life. I get to work with amazing people and think about amazing things every day. If someone out there is wondering if it is worth it, from my perspective it certainly is, even in the current economic climate. At the end of the day, there are plenty of scientific problems out there to solve, and it shouldn't be too hard for well-trained and motivated people to find a niche. So yeah, I can't really complain. However, the topic of training too many PhD students keeps coming up, so I thought I would drop an idea. One solution could be an NIH and NSF mandate to start paying PhD students and postdocs more. If you were paying students and postdocs twice as much, you would only be able to hire half as many, and the quality of the students recruited would probably improve. Why not use some of that NIH money to sweeten the pot for potential grad students, rather than building another building or hiring another administrator? You need quality, not quantity, after all, and you generally get what you pay for. There is huge pressure on my generation to achieve personal financial stability. Thus, you are potentially losing some of the best scientific minds to medical school, Wall Street, and other financially attractive paths. Imagine if the person who would have figured out a universal flu vaccine has just been recruited to a consulting firm. Nothing against those kinds of jobs, but that would be a very inefficient use of talent in the bigger scheme of things. It is mostly the idealistic goofballs like myself that are going into science, and given the circumstances, I can't blame others for choosing a different route. The blood, sweat, and tears of PhD students and postdocs are what keep a large part of the scientific progress in this country moving. Pay them well, scientific quality will improve, and job saturation will go down. Figuring out how to do this from an administrative or political perspective isn't trivial, but it seems like an idea that should be seriously considered. I'd love to hear your thoughts. The weather in Salt Lake City is a sunny 69 degrees Fahrenheit. Again, thanks for the show and keep up the good work. Cheers, Justin. And then he has a listener pick. Should we do that later?
Yeah, we could do it later. If I remind me, I'll forget.
Yes. Well, I mean, yeah. Okay.
I will forget, and you'll remind me both.
Yeah, right. Right.
I think it's a good idea to pay, sure, students and postdocs more. I don't know how you do it. And any thoughts? I mean, you can't just say I'm going to pay students twice as much. I mean, we're sort of limited here as to how much you can pay them by NIH. So it has to come from NIH.
Right.
I don't think it's a good idea at all.
No?
What you have, there's the problem, one of the big problems in research right now is that there is a vast glut of labor, right? You've got way too many PhDs competing for too few jobs. Mm-hmm. I think there's pretty broad agreement on that now. And this is a problem that's been brewing for at least 30 years. The fundamental issue here is that the training and the labor force were linked at a time when it made sense to do that, and it stopped making sense to do that once all of the jobs were filled and the funding was flattening. So we no longer have the post-World War II boom and vast expansion of science sopping up all the PhDs we can produce, but we still have this system that links the training to the labor pool. So the only way to run a lab economically is to use the cheap, skilled labor of graduate students and postdocs, right? And that's the problem. We don't. This is a very circuitous way. Doubling the pay of graduate students and postdocs, yes, you could only employ half as many, but it's a very circuitous way of getting at the problem. A much more direct route would be to eliminate the pay of graduate students and limit postdocs to like 1 per R01 grant. So just to phase this in over a period of time, obviously, otherwise you would utterly destroy the system.
Alan, are you suggesting training grants only?
What I'm suggesting is more of the training model that is used for medicine. Okay, so look at, and I wouldn't normally point to the American healthcare system as a good example of anything managerial, but this is one case where it functions more or less correctly. If you look at what the Department of Health and Human Services does in managing medical training, they have a lot of rules about it and a lot of regulations, but they don't pay stipends to medical students.
Some
right?
do. Some do.
There are scholarships.
No, the rich medical schools pay stipends.
The medical schools, that's who's paying.
Yeah.
The rich medical schools
Right.
offer scholarships and will pay the students who are able to get into those scholarships.
And the MD PhD program goes free.
The MD PhD program, because it's financed through the PhD.
That's true. That is true.
But look at what happens. There is absolutely no shortage of applicants for medical schools. Right?
True.
They're taking a
true.
tiny percentage, even the middle of nowhere medical schools are taking a tiny percentage of applicants.
Correct.
Everybody wants to get in because they know if they manage to get in and they complete the 4 years of medical school and then they go on to a residency, which is the medical equivalent of a postdoc, after they've finished that amount of training, yes, they've taken a huge financial hit from medical school. They've probably gone deeply into debt, and then they've lived at below market wages for their skilled labor as residents for a few years, but after that, they are essentially guaranteed employment. Now, it's not as well paid by any stretch as it used to be in real terms. I'm intimately familiar with that, but it is a fact that if you have a medical degree and you've completed residency, you can get a job. And you can get a job in most specialties of medicine that will pay a decent wage. You won't get rich doing it, but you won't go broke either. So the labor market there is functioning effectively. It has a huge barrier to entry, which is the medical school admission process, but it is worth going through that, and it's worth paying your own way through that, because at the other end, there's a job.
I wonder if you'll get the same, the same level of interest in graduate work as you do in medicine. I mean, there's just these gobs of people who want to be a doctor.
Yep.
Yes.
Yep.
And you will have.
Tell me about that. I serve on our admissions committee too much.
Yeah, but the people who really, really want to be scientists are the ones you want being scientists.
Yeah,
anyway.
absolutely.
There have to be jobs for them.
That's why they have to
There
be
have
paid
to be
more.
jobs for them, and the way to ensure that there are jobs for them is to produce fewer of them. The way to produce fewer of them is not to pay them more to do the training. That's the way you draw more people into the training.
Well, no, not necessarily. Not the way that Justin was proposing. Is that if there's a fixed amount of money and you're now spending it on half as many people, then you are going to be more selective. And
Just admit half as many people.
Yeah, we take too many. I agree.
You've got
We
too
take
many PhDs in the program, and obviously equity is an issue. That you're going to have people from certain backgrounds are going to have a lot easier time affording their own way through graduate school without a stipend. But that's an entirely fixable issue. Again, pointing to the medical school model, you can have scholarships for people who need them, and you can have scholarships for people who have merit-based admissions. But it should be the training should be something that is worth doing and worth investing in from the perspective of the student. And if it's not, then that tells you your labor pool is glutted and it will correct itself.
Part of the problem, Alan, is that a job as a doc is a clear job. You have a goal. There are things you take care of people, right?
Yes.
When you do science, you figure out on your own what you want to do, and not everyone is good at doing that. So it's not like you just walk into a lab and will be successful like a physician will be successful. So
you figure out on your own what you want to do, but the job description of a PI is actually a fairly obvious thing. You are going to get grant support to do science. That's what you do.
Yeah, it's just not
the nature of the science. I agree that the nature of the science is much more flexible than medicine, which is a procedure. But the jobs themselves have very well-defined...
I agree. I totally agree, but the outcome is not guaranteed.
No, the outcome is not guaranteed, and then that's already accounted for in the grant process.
So you take care of a patient, you get paid no matter what, but you may not figure out...
you might get paid no matter what.
You should be. But if you go in the lab and you may not do the right experiments, you won't get funded, and that's it, right? So that's why it's going to be always less attractive than...
I am not advocating that we adopt the financial system of medicine for science. We already have a system for science where it's funded directly by grants from the government. That is not the system we have for medicine.
Yeah.
The system we have for medicine, well, let's not even get into that. And it's not guaranteed as a doctor that you'll get paid, by the way. But the job is there, and somebody will eventually cut you a check for the work that you did, even if the hospital is eventually going to go bankrupt. So that's a further downstream issue, but I think in terms of fixing the labor problem, what you need to do is turn off the tap, and the way you turn off the tap is to put fewer people into the program. And if you limit the population of postdocs and you do away with this whole business where the NIH force feeds trainees in and pays for them, then that will limit the population of graduate students. And how do you staff your lab? Well, you staff it with technicians and senior scientists. And those are going to be the jobs that will be available to the people who complete this training.
Yeah, I think that the senior scientist idea, which we've talked about, is an important component of this, right? But I think you have to train some student. You have to cut it down to a certain point, because you do need future scientists, right?
You need at least an order of magnitude reduction in the number of scientists we're training. And it's going to take radical action to accomplish that. So I'm sure we'll get mail about this.
That's fine. That's what we're here for, to get mail. All right, let's do a couple more. Rich, you want to take the next one?
Jennifer writes, "Greetings, denizens of TWiV. Currently sunny and around 65 here in Silicon Valley, though we thankfully had a bit of rain this morning for the first time in a while. I am currently an undergrad at San Jose State University, daydreaming about studying viruses in graduate school and trying to learn as much as possible. I just finished listening to TWiV 326 about soluble HIV antigens used to protect against infection." I know, I'm late. "And I remember someone commented on how this wouldn't be an effective treatment because for HIV because it can't eliminate reservoirs. While this is true on its own, couldn't it be combined with other therapies such as HDAC inhibitors to activate the virus? As the virus is activated, the immune system can eliminate those cells that are infected and soluble antigen." Does she mean antigen, or is this antibody?
No, these are CD4, basic soluble CD4.
Ah, right. Soluble antigen would prevent the viruses from infecting new cells and potentially forming new reservoirs. Eventually, the remaining reservoirs would be eliminated and no new ones would form. Though several studies have shown that these reactivated reservoirs are only poorly eliminated by CD8 cells in patients anyway, so that would have to be addressed as well. I guess what it comes down to is whether this new method is more or less effective than HART at preventing virus from infecting new cells. What are your thoughts? I think it's a really exciting possibility for treatment in any case and may resolve the many issues with vaccines that currently plague the field. I really hope it pans out on further trials. Keep up the good fight. Love the show. Thanks."
Yeah, so people are trying this. For sure. You activate it with these histone deacetylase inhibitors, turn on transcription, and then hit them with heart or hit them with whatever you got. Could be soluble CD4 for sure.
It's a way of bringing the latently infected cells out in the open so you can kill them.
I asked Steve Goff, and he said, yeah, but the problem is you're never going to activate every last provirus. There's always going to be one, and then that'll get you in the end. So he's not very enthusiastic about it. But it's being trialed for sure. Absolutely. Let's see. Anthony sent a link to a CDC article, Emerging Infectious Diseases article about influenza A, H7N9 virus transmission between finches and poultry. And he writes, this is particularly bad news because the wild form of the model animal used in this research, Lonchura striata, is widely found in the Chinese prayer bird trade. Many are transported around China without documentation. The birds are purchased for release as part of cultural ceremonies. So
basically, these birds can vector this nasty virus to poultry.
Right.
And mostly through the water, from what the abstract said.
All right. Let me just take the next quick one, and then we can get you guys to do some. Steve writes us, he sends a report from the UK Food Standards Agency about viruses in the food chain. It's quite a nice report. I've never seen one of these before. So these are reports put out over in the UK, and Steve is a listener who we've heard from before. And so check this one out. This is pretty cool. Alan, can you take the next one?
Okay. We're down to Pete.
Yep.
Pete or Piet. Dear Vincent, Dick, and Alan, I have recently discovered your podcast by randomly strolling the web and have found it to be an inspirational experience. I'm only at episode 53 at the moment, so lots to listen to. I currently live near Cape Town in South Africa and listen to the podcast during my 1 hour and 30-minute drive to work and found that I enjoy traffic more and more now. I'm a finance math major that has found my way into computational biology and currently work for a biotech company in Cape Town. I will soon be traveling to the States to start my PhD at Oak Ridge National Lab in the fall. Congratulations. Your podcast has inspired me to focus my research more into studying viruses from a bioinformatics point of view. I will also be working on plant microbe interactions and bacteriophages. Just wanted to say thanks for the very entertaining, humorous, and intellectually stimulating show. I also found this news article online that I believe might be of interest to other listeners, given the recent Ebola panic. And sends a link, which I'm just looking at now. Yes, this is the paper that we just covered.
Yes.
So when you get up to this episode in a month or so, Pete, you should really enjoy this.
Dickson, it's perfect that you should get this next one.
You think?
Yeah.
Okay, Gary writes, hi, gang. I was just listening again to TWiV 147 with just Alan and Rich. And I have listened to many others where you talk about GMOs and how there's nothing wrong or you are undecided about them. While I am a huge supporter of science and in general do not have any problems with most things, I do have a problem with GMOs. First, no one can tell me that it is safe for me to eat pesticides that were somehow baked into crops. If the pesticide is not able to be drunk or even eaten in any other form, why would it suddenly be okay to eat it just because it was somehow engineered into a plant? Common sense tells you that it is not safe. I am 100% comfortable with the idea of GMOs that add vitamins or minerals. I am thinking about the rice that adds vitamin A, maybe. Can't remember exactly which it adds. I do think, start again, I do not trust Big Ag, but that's me. I understand that you guys must trust them, though, and I honestly do not know why. I am not a big fan of drug companies, but that is mostly because of business reasons. Why would drug company A make a vaccine that stops someone from getting HIV when they make billions on treatments for said virus? Just wondering. I do love the show because it makes me think about these things. I am now more willing to listen to drug companies. While I have never been against vaccines, your reasoned arguments in favor of them have always been useful when talking to anti-vaxxers. I realize that this has bounced around a lot, but I have not written before and have had many of these thoughts just trying to get out. Love the show and have worked my way through the backlog once and going through again to make certain that I understand fully. Gary.
Back catalog, not backlog.
Well,
that makes it sound bad, Dickson.
Oh, okay. I don't want to make it sound bad. That would be the last thing I would want.
Tell us about GMOs, Dickson.
Well, GMOs, it's another term for genetic modification. So either you're against lab work or you're not.
But Dickson, we've talked about this on TWiV and putting things into plants. So for example, there's stuff you eat and then there's plants that you don't eat, right?
Right.
So you can modify the plants you don't eat, and shouldn't
so
be an issue. But although there are concerns about the genes you put in getting into the environment, you have to be cognizant of what they may do.
That's a concern.
Now, he's talking about putting pesticides in food crops. What is this about, Dickson?
Well, it probably relates to BT, Bacillus thuringiensis protein, which is an antimicrobial agent that forms a crystalline structure in the gut tract of an insect after it eats the plant. So we eat that too. Now, is that bad for us? Because we have an acidic gut. But our gut is a lot larger in diameter than most insects' guts. And as the result, when this crystal forms inside the gut of an insect, it blocks it up and it actually creates a constipation-like syndrome in the insect. When they crystallize in our intestinal wall, they're probably digested by our proteases and don't cause us any harm whatsoever. So eating that kind of a genetically modified crop plant, like tomatoes, for instance, as they engineered in Israel, would not be harmful at all. In fact, they test these things out a lot in animals before they actually release them to the environment. But the thing I'm against in GMOs is to engineer a plant to resist more, let's say, herbicide like glyphosate.
Yeah.
Like glyphosate is made by Monsanto. And so Monsanto has now realized that the weeds have become as resistant to the glyphosate as the corn plants. So now they have to re-engineer the corn plants to be another level above that. And that's really a bad use of genetic modification, in my view. That is strictly for getting more money for it. Not only do you have to buy more weed killer, but you have to buy the genetically modified corn plants in order for them to resist that that herbicide as well. So if it's for a nutritional reason or if it's for a general public health reason that you're trying to improve the nutritional status of a plant by improving its content, be it vitamin A or vitamin B12 or amino acids, cassettes of amino acids, I think that's all to the good because you already eat those things anyway in your diet. We're just trying to give you more of them in a smaller source. And so the International Rice Institute in the Philippines is doing a wonderful job of making rice more of a single food than it already is. That's basically what they're trying to do. And for other plants, too, because people tend not to eat a wide variety of plants if they live in, let's say, on the edge of poverty, for instance. And rice is a staple diet that they supplement with very little protein. So if you could make it into a more protein-like plant, as they do in, let's say, Louisiana, if you're not going to this meeting in New Orleans, you're going to miss out on red beans and rice. So you eat red beans, you don't get enough. You eat rice, you don't get enough amino acids. But if you eat them both together, you get a perfect food in terms of the amount of required amino acids that are necessary for your diet. So if you could engineer beans or rice to have all 9 of them, then that would be a good thing, I think. So.
I don't know what 147 was not just Rich and Alan, so I don't know what you guys said that set off Gary.
But it's a huge topic because I think there's a lot of pushback from environmental groups, particularly in England, particularly in Europe, that suggests strongly that they're all against Frankenfood. They call it Frankenfood, but it's really a misnomer because the book Frankenstein's Monster was the title, not Frankenstein. So it's Frankenstein's food. No, it's not. I don't even know what to call it, but it's a silly thing to.
There's a big parcel of issues here.
Yeah, huge.
A lot of the international angst about these is very early on, since the U.S. embraced genetically modified crops sooner than a lot of European countries.
Yeah, that's true.
This became a very convenient way to have a non-tariff trade barrier that was not covered by any of the existing treaties,
that might be right.
so that you could stop imported inexpensive rice from the United States from coming into Europe, so that your French farmers would not throw up barricades in the street because they were being put out of business. So there's that whole issue. That's what got a lot of farmers in Europe on the anti-GMO bandwagon. It had absolutely nothing to do with the science. I have not seen any really solid scientific objections to these things, largely because humans have been genetically modifying our food plants for 40,000 years.
That's right.
There is no crop currently cultivated that is not genetically modified.
Right.
And by the way, the most popular pesticide among organic farmers is BT.
Right.
They spray the stuff all over your organic farm stand food that you're buying and eating. So, Gary, you're already eating this pesticide, and it's not killed you yet, and it's not going to kill you.
It won't do it.
I'm absolutely fine with that. And by having these BT transgenic crops, you have to understand the alternative is not organic agriculture feeding a planet of 7 billion people. It can't.
Yeah, that's right.
If you want to feed 7 billion people, you need to do something about the bug problem that's more aggressive than what's possible in organic agriculture. So the alternative to something like BT transgenic corn is not organically grown corn. The alternative is corn that's been sprayed with malathion and all the rest of the nasty stuff that gets sprayed that kills not only the insects that eat the corn, but also kills every other arthropod in the vicinity and downstream. So environmentally, the effect of BT transgenic crops has actually been millions and millions of liters of petrochemical pesticides not being sprayed on fields. And I consider that a huge win. And these foods, no, they're not harmful. If you want to think of a harmful cultivated food, look up cassava. Okay, that is some dangerous, dangerous stuff. Nobody's genetically modified it, but if you pick up your cassava root from the field and you pop it in your mouth and you start eating it, you are making a trip to the emergency room.
Is that cyanide by any chance?
That is cyanide, and that is a staple crop for hundreds of millions of people around the world who subsist on that as their primary nutrition source.
That's true.
So this is a non-issue, I think.
All right. Any other thoughts?
People have a tendency to, GMO's just gotten a bad rap, and people, without understanding the science, just say that it's bad. And as Alan has pointed out, there's nothing intrinsically wrong with it. As Dickson points out, like any technology, it can be used properly or it can be misused. So the key here is to understand exactly what you're talking about and make a judgment based on the science.
Sure.
Yeah. I think blanket condemnations are
inappropriate,
right? Like saying you don't like cherry trees. How can anyone do that?
We have no idea. Or angiosperms in general.
A what? Let's do some picks of the week. Alan, what you got?
Okay, I have a bookmarking site. Now, of course, you've heard about these sites that offer bookmarking services, I'm sure. The social bookmarking, Delicious was one for a while till they went under. This is anti-social bookmarking. So I ever since Delicious went under, I've wanted a place where I could store my bookmarks online, because invariably I'll be looking at something and I'll have my phone and I'll be looking something up. I'll say, oh, I bookmarked that on my desktop, or is it on my laptop? Or it's just so handy to have these online, but all the social bookmarking sites are the usual internet deal where they'll give you some minor service for free so that they can sell you to advertisers, and ultimately, this is just not sustainable or really tolerable. So my pick of the week is Pinboard, and pinboard.in is their address. It's a business model that your grandparents would recognize. You pay an annual fee. So it's currently $11, and you put your bookmarks on the site. And if you want to make them open to the public, you can make them open to the public. If you want to keep them private, you keep them private. There are no ads. There's no spam, because the spammers are not going to pay $11 to subscribe to the service. They've got all kinds of neat little. You can bookmark things by email. You can do neat little stuff like that. It's just a very simple interface, very straightforward, and I love it.
Your modern browsers will store your bookmarks in the cloud. You can access them anywhere, right?
If it works properly, and as long as they are able to maintain that business model sustainably, and
Well, Google is going to be around for a while. Safari is going to be around.
And what is Google doing with your data? I mean, they're feeding you ads based on that, and then they may decide they're going to cancel the service if it's not profitable.
Okay, well, they can look at my bookmarks. I don't mind.
Oh, I don't care if people look at my bookmarks. What I care is that the site is not cluttered up with a bunch of cruft.
Right. Okay, got it.
So anyway, I love this.
Do you like cherry tree blossoms?
No,
Okay, good.
they're okay.
Wow.
They can be.
Rich Condit, what do you have?
So we talk a lot here about the academic institutions being addicted to NIH funding and other government funds through indirect costs and that kind of stuff. And on occasion, I have this discussion with my chair, who has pointed out to me that no, this is not necessarily the case, or it's not necessarily the case that the academic institutions are making money off of indirect costs. And he pointed out to me an article that's American Association of Medical Colleges publication, AAMC, titled Academic Medicine Investment in Medical Research. And this is a study done by the AAMC, who hired a consulting group called Huron to question a bunch of different medical schools. They did 46 institutions receiving between $26 and $750 million in external funding in 2013, and they had a big, question, form for them to fill out. And the bottom line is that the average medical school investment was an additional 53 cents for each dollar of sponsored research received. So the bottom line is that, yeah, they're getting a lot of indirect costs, but they're in order to really adequately support the research enterprise, they're putting in more money. It amounts to an average of $111 million per medical school. I had heard this before, but I'd never seen it formalized. I had a friend at another institution that won't be named who said that their dean was talking about cutting back on basic research because it was too expensive.
Yeah.
So I'm just throwing this out there as a counterpoint to that argument that we often talk about. I thought it was an interesting article.
Do consider the source, though.
Oh, yeah.
The
AAMC. Sure, they have an interest in this.
Yeah, yes. I mean, this is the lobbying group for the medical schools. So they're, right. And this is a phenomenon where the accounting is exceedingly complex, and it would probably be quite easy to read the numbers in your preferred way. Just saying.
And
I'm not saying that's what happened.
It's 53 cents on the dollar is not enough. It probably doesn't. I mean, medical schools don't pay much of our salaries, right? That's traditionally. Right. So that needs to change. It really needs to go up. I appreciate $100 million per medical school, but that's not a lot in terms of their budget. They need to invest in their faculty. If they want basic research, they have to invest in it. It's just not clear to me that it's something they want to do, unless it gets them a Nobel Prize, of course, in which case they're happy to do it. Kathy, what do you have?
I have something completely light compared to all of these things. As one of my Japanese colleagues said, this is a nice kind of paperwork. So it's a Japanese paper company site or link to their company's ad. I don't know what it is. Anyway, the person created it for this paper manufacturer. It's tissue paper stop motion animation. And so you can see some still photos of these various things that they folded out of tissue paper. And then there's a video link in there for a YouTube-like video. And then you can go to the end and see how they made it. And they're just amazing. So I highly recommend taking a look at these for something completely different. They're not quite like origami because they're made out of tissue paper, so they have a softer look.
All right, these are cool.
Yeah, I looked at these. They're cool.
I could look at these. Yeah, they're okay.
Yeah. Well, check out the video.
Just don't look at the cherry blossom ones. That's right.
Hey, Dickson, what do you have for us?
Well, I have another article that I cut out of this week's Science magazine. It's an article called Research Funding, and the title reads, Big Names or Big Ideas? Do peer review panels select the best science proposals? Mm-hmm. And it's written by two, I guess they're sociologists. One is in the National Bureau of Economic Research at Cambridge, Massachusetts, and at Harvard. Let me just read the abstract because this is worth listening to. This is something we may not have been able to predict. Ha ha ha. So this may be overstating the obvious. This paper examines the success of peer review panels in predicting the future quality of proposed research. we construct new data to track publication, citation, and patenting outcomes associated with more than 130,000 research grants, projects, R01s, funded by the U.S. National Institutes of Health from 1980 to 2008. We find that better peer review scores are consistently associated with better research outcomes, and that this relationship persists even when we include detailed controls for an investigator's publication history, grant history, institutional affiliations, career stage, and degree types. A 1 standard deviation worse peer review score among awarded grants is associated with 15% fewer citations, 7% fewer publications, 19% fewer high-impact publications, and 14% fewer follow-on patents. Period. Ain't that interesting?
You know, it's so rarefied at the top of the score that this is almost a given, right?
Yeah, I mean, if you get a 1.0, I only got one of those in my whole life. I don't know if anybody else here got one of those, but I got one of those just once. My publication rate skyrocketed. No, that isn't true at all, but it should have perhaps. But I think this is an interesting correlation, but one could have guessed this, having served on a peer review committee. I mean, I think that we all feel this way.
Part of the problem is that the ones that get the highest scores have a lot of the data already done.
But of course.
Right?
Yeah.
And it's a sure thing. But the ones that are a little bit risky, forget it.
You mean like a self-fulfilling prophecy, so to speak?
Yes.
Yeah, that's right.
In a way.
That's right.
It's an interesting article, though.
Yeah, I thought you'd enjoy it.
I have a blog post by Steve Novella, who writes, he's a doc up at Yale, who writes interesting posts over at Science-Based Medicine, and a new study on the potential or whether there's an association between measles, mumps, rubella vaccine and autism. Just came out, and he writes about this and summarizes it. It's 95,000 children. Oh my gosh. And, there's no evidence that MMR does anything.
But you know what does have some correlation here, Vincent? Sorry to interrupt. My wife pointed this out to me.
What?
Older women and older men having children have an increased rate of autism.
Yes.
Mm-hmm. Yeah,
now I don't know how to explain any of that data, but that's the truth.
Yeah.
Anyway,
that's remarkable.
This is I could believe that, but not MMR. It's clearly and Novella basically writes, we probably didn't need to do this, and we shouldn't do it anymore, but we probably will. So,
right, but this was sort of taking another angle on it. They looked specifically at kids who had older siblings with autism and kids who had older siblings without autism, just to see if, in yet another population, maybe a population with increased risk, maybe you would see some effect, and you don't.
Yeah.
I like his paragraph toward the end. It seems to me that if we're going to spend limited resources to study a question in order to reassure a public concern, then we should endeavor to ascertain whether or not the concern is eased by the research.
Yes.
History indicates that it largely isn't.
Right.
One possible solution to this dilemma is to tie such research funding to specific regulatory measures.
Yeah.
So they say, for instance, research into vaccine safety above and beyond what is scientifically necessary could be tied to laws removing vaccine exemptions if those vaccines are found to be safe.
Right. Yep. So we have a couple of listener picks. Ramon, who wrote us earlier, sent his pick as the actual article that Novella is writing about. It's published in JAMA. Autism Occurrence by MMR Vaccine Status Among U.S. Children with Older Siblings and With and Without Autism. So thank you, Ramon. Mehul Suthar, your friend from Emory, right, Kathy?
Mm-hmm.
He sent a link to a Coursera course on Ebolavirus disease. So if you want to learn all about it, you can check that out.
It runs through May 11th. Now, you know more about Coursera than I do, Vincent. If May 11th comes and goes, is it too late?
Well, the way they work is they have a paced course until May 11th. Every week they release material, and then you get a quiz every week, right? Mm-hmm. So you can still view them after May 11th, but you won't have the quizzes, you won't have the pacing, and so forth. So my two courses are still up there, but, you can't take most people do it that way.
Okay.
Justin had a listener pick. He's picking Molecular Biology of Bacterial Viruses by Gunther Stent, an oldie but certainly a goodie. It was It was recommended to me by one of my committee members when I first started as a PhD student. It covers a lot about the old school bacteriophage experiments that are still very relevant and useful to think about. Every time I read this book, it gets the gears in my head turning. It's a bit heavy, but crucial reading if you happen to be interested in phage. Yeah, this is a good book. I have it on my shelf as well. Used to be one of the ones you always read in grad school.
Mm-hmm.
All right. Thank you for those, Ramon and Mehul and Justin. And that's TWiV335. You can find it at iTunes and TWiV.tv. We'd like to get your questions and comments. TWiV at TWiV.tv. Dickson Despommier is right here at Columbia, also at verticalfarm.com. Thank you, Dickson.
You're welcome. This was fun.
Yes, you were animated today.
Well, I felt turned on.
I'm not going there. Kathy Spindler is at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This was fun. I enjoyed reading these papers.
Rich Condit is at the University of Florida in Gainesville. Thank you, Rich.
Sure thing. Always a good time.
Alan Dove is at turbidplaque.com. You can also find him on Twitter at Alan Dove. Thank you, Alan.
Always a pleasure.
I'm Vincent Racaniello. You can find me at virology.ws. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.